Okay, so good morning, everyone, and welcome to the fourth edition of our webinar of the South American Initiative for Molecular Simulation. Today you have as guest Denys Santos from the Federal University of
Good morning.
Pernambuco here in Brazil. So he'll talk a little bit about his work. So please, Denys.
Yeah, thank you very much for the
You can
invitation.
share your screen.
Yeah, of course. Thank you very much. So let's start with the presentation. Let me see. Okay, so now probably you'll see.
Yes.
Okay. So I'm going to start. If something goes wrong, so just tell me. I'm not seeing you.
Yeah,
So
sure.
yeah, okay. So good morning, everyone. It's a pleasure to be here to talk a little bit about the work I'm developing during my PhD. Oh, something has gone wrong. Okay, I go. And I'm going to talk about Suave. That is a program that I'm developing since the beginning of my PhD, and is a program that we use to calculate properties, geometrical and physical properties of biological systems, chemical interfaces, taking into account their morphology. So the interest for calculating or for developing such a program started in 2017. During this year, I was studying the interaction between an antimicrobial peptide, the polymyxin B, and different chemotypes of gram-negative bacteria, Pseudomonas aeruginosa. And during the studies, we were studying the interaction between the outer membrane of this gram-negative bacteria and the antimicrobial peptide. It was quite interesting to see that according to the susceptibility of a chemotype, a different chemotype of this Pseudomonas aeruginosa, it was possible to form a curvature in the system. So if the chemotype is very susceptible to this antimicrobial peptide, there was a formation, a highly curvature formation in the membrane. So this was very interesting to see because we could see exactly what is expected from experimental results. And it's important to calculate the geometrical and physical properties of this membrane during this process of the curvature formation. The problem is that when we are using usual programs for simulations, they are not able to calculate, for example, the area per lipid or the thickness or the volume of this bilayer, taking into account the morphology, or they are not able to calculate chemical interfaces, and we have seen some different approaches. All of them basically are using Voronoi algorithms. Then for phospholipid membranes, these algorithms are just taking the phosphate groups for the membranes and then using these acids to make a Voronoi tessellation. And with this Voronoi tessellation, these programs are able to calculate area per lipid for the most different types of membranes. It's just a problem when these programs are just using this Voronoi tessellation, but they are projecting the Voronoi polygons onto the XY plane. And with this, they are like losing information about the curvature of the system. So even they are using the Voronoi polygons to calculate the surface of the membranes, they are projecting this onto the XY plane, and with this, a lot of information is lost. So this is exactly the problem we are trying to solve when we are trying to calculate geometrical properties for systems with different morphologies that are not flat. So to do so, we have developed Suave. So in few words and in few pictures, I have here how Suave works. And then if you just insert in Suave a PDB file that is a structure file for your system, for your simulation, trajectory file in a PDB format, you can select some atoms so that Suave can use these atoms to drive a fitting procedure. And with this fitting procedure, we can generate the fitting surface. And with this fitting surface, we can calculate any type of geometrical and physical properties you want, taking into account the morphology of the systems. It's important to see that the heart of this procedure is the fitting procedure, is the fitting process, the third step. So to calculate the perfect fitting, we have used different fitting surfaces, different fitting procedures. We have started with the modified Lagrange interpolation. That is a quite nice procedure to interpolate the surface, a bidimensional Lagrange interpolation. But there's a problem with this interpolation because as this is an interpolation, it is just bringing the fluctuation of the phosphates along the simulation to the measurements we are making. For example, if you just interpolate the surface, bidimensional surface, and the atoms are fluctuating along the simulation, and then you measure the area per lipid of this membrane along the simulation, you'll see a noise because of these fluctuations of the atoms. So this is nice because you can interpolate the surface, but you are increasing the noise, the response, you are increasing the noise in the measurement. So this is not exactly the best solution. So we have used a different one after this, that is the inverse distance weighting procedure. So we are actually making an average, a weighted average on the position of the atoms, like phosphates in case of membranes and phospholipid membranes. And with this, we are using as a weighting function, a radial basis function that is using much more the contributions of atoms nearby. One point we are going to set in the surface, we are trying to fit. And with this, we are not considering atoms that are far apart. So with this, we can not exactly make an interpolation, but it's almost a fitting for the surface. And we can actually calculate this for three-dimensional, four-dimensional surface. This procedure is very useful for any type of fitting procedures. So the first step, the first run of this procedure, we have used the inverse hyperbolic cosine as the radial basis functions, but actually it's not so practical when we are trying to differentiate and integrate this type of function. So we have used another one that is a Gaussian function, and with this Gaussian function as a weighting function, it was possible to insert a parameter that is the width of the Gaussian function. And with this width of the Gaussian function, we can create a dependence on the number of sampling points we are using to drive the fitting process. So this is exactly what we are now using, and we have published exactly this kind of solution in the first paper of SUAVE. And I'm going to describe you how to calculate the best fitting parameter that controls the width of the Gaussian function, and how we can use this to make the best fitting for a bidimensional surface, and then how to apply this for some systems that I'm going to show you. So the advantage of using, of course, the surface that we are fitting is, of course, that we can calculate the gradient, differentiate the surface, we can calculate integrals and the curvature. What we need to do so is just the fitting parameters and a reasonable amount of sampling points. I'm going to show you now the fitting parameter, and then after I'm going to show how is the dependence of the number of points from the surface in relation to the fitting surface we are trying to fit. So to understand how the fitting parameter depends on the density of points, the number of points we are using from the interface we are trying to fit, we have used different surfaces and in different number of points from the surfaces. We have started with densities like 0.36 atoms per nanometer square, and we have used up to 900 atoms or points per nanometer square. So in this range, probably all chemical interface can be between these two limiting numbers. So as you can see here, for each density profile, there is an alpha parameter, there is an alpha number that minimizes the RMSD between the fitted surface that we are trying to build by using SUAVE and the sampling surface. And then using these minimum points, the points that minimize the RMSD between the fitted surface and with the sampling surface, it's possible to see in the dashed lines a profile that describes perfectly what is going on with the alpha parameter in relation to the density of points from the surface. So with this, we can see exactly a profile that I can show you here. The black points in this profile are the points that minimize the RMSD, and in red we see exactly the profile that I'm of the curve that I'm showing here, the alpha, depending on the density multiplied by 0.742 and so on. So with this function, it's possible to calculate the best width for the Gaussian function that is weighting the contributions for the points on the surface. So once we have set these, it's important to see how, even if we are using the best parameter for the fitting, we just have to see how the density of points interfere in the building of the interface, in the building of the fitting surface. So this is one example on how many sampling points do we need to make a best fitting interface. So we have used this very strange blue sampling surface just to see if Suave can calculate, can fit exactly where this surface, how this surface is, and all the fluctuations of the surface. And we have used such a small quantity of points, like 0.36 points per nanometer square, and then we have used up to 900 as in the previous analysis when we were evaluating the alpha parameter. And as you can see, if you are using a low quantity of points, a poor quantity of points, you can see that there is not a good relation between the sampling point and the surface we are trying to fit. So these two surfaces has nothing to do with each other. But when we are increasing the number of points from the sampling surface, you can see that the representation Suave is making for the surface is increasing. The representation is better than the previous ones. And if you increase to 2.41 points per nanometer square, you see that probably the representation made by Suave is almost the sampling surface. And even better if you continue to increasing the number of points from the sampling surface. So as you can see here, from 26 points per nanometer square, the RMSD between the sampling surface that is in blue and the surface Suave is building that is in red, you can see that the RMSD between these two vanishes. So this is nice because we can calculate exactly what is going on with the surface, just evaluating the surface Suave has built according to the sampling points from this sampling surface. So this is for an open surface, but actually we can do that for a closed surface. Here you can see an example of a very strange closed surface, but it's perfect to see if Suave is able to calculate such a strange, can fit such a strange surface. Suave is able to calculate more easier functions, not changing this, not having this such a strange morphology. So we have used a few quantity points like 0.114 points per nanometer square to calculate the fitting function, and then we have increased the number the same way we have done in the previous slide. And as you can see, when we use 8 or 32 points per nanometer square, the fitted surface is almost exactly like the sampling surface we are trying to fit. So in this point, the RMSD between these two surfaces vanishes. So the conclusion here is that once you have a reasonable amount of sampling points from Suave, you provided this to Suave, you see as
So, hi guys. I believe that we had some crash with Denys. He's not online anymore. Okay, so we just wait a few seconds to see if he will return. So yeah, today is the day to have issues with the internet. So we just add to the stream our second guest. Hi, Nina.
Hi, everybody.
Yeah, so this is Professor Nina Pastor from the Universidade Autônoma del Estado de Morelos in Mexico. So, okay, Nina, I believe that you can share your screen and we can see. I'm still waiting. If Denys did not return in 1 or 2 minutes, so we can start with your talk.
Let's see how this works.
Yeah, just share screen and then I will add this to the streaming.
Okay. I'm gonna show everything.
Yeah.
I don't know if, can you see the presentation?
Yeah, here it is. Okay.
Okay, so
Yeah,
so I start when you tell me.
yeah, I believe that you can put your presentation on and start, since we are not having any news from Denys. So we are sorry, guys. So we have this problem with the Denys connection. So we start now with the talk by Professor Nina, and then later when Denys return, he can finish her talk. So thank you very much, Nina, for accepting the invitation to give us a talk, and please feel free to speak.
All right. Thanks for allowing me to join this South American initiative. Talking with Sergio Pantano, we decided that South America starts south of the US-Mexico border, so I'm officially South American too. Now, from what I was listening to Denys, this is a completely different story. I am a biomedical scientist, so I am very worried about biological stuff. In particular, I'm interested in structure and dynamics of proteins, and we know that these are inherently intertwined. So even if you have a perfectly folded protein, or if you have a disordered protein, you're going to have motion, and these motions are characterized by different both time and length scales. So when you work with folded proteins, normally what you care about are small fluctuations, especially on short time scales, like say up to nanoseconds. But if you're working with a disordered protein, then you also have slithering motions, and those can happen up to the millisecond rate, time scale. In either case, one should always be aware that proteins have to be represented as statistical ensembles, not as fixed structures, and that these are very heterogeneous collection of conformational states. It becomes really interesting from the biological point of view because interactions with other molecules will change the equilibrium amongst the states that form these statistical ensembles. And this is, in a nutshell, information transfer, because when the protein changes conformation, it presents a different molecular surface with which it can interact with other proteins, and then information is transferred across signaling networks inside the cell. In particular, when one is dealing with disordered proteins, it is very important to know how it looks like before and after the interaction, and also to care about recognition and specificity mechanisms. For a particular collection of disordered proteins, Upon forming an interaction with its partner, there can be disorder-to-order transitions. These happen at particular regions of the protein, which are called morphs, or molecular recognition features. Depending on the conformation that the peptide acquires upon binding, they are classified as alpha morphs if they form alpha helices, beta morphs if these are extended structures like beta strands, iota morphs if they are irregular, and complex if they are a combination of any of the other. It becomes even more interesting if, even upon binding, there is no structuring. So these are called fuzzy complexes. So I tend to imagine this. For these, you cannot get a picture. There's no way to crystallize them or even to look at them through cryo-EM because they don't keep still. So they remind me of an octopus and its tentacles grabbing things. So it doesn't really matter which sucker it uses, just as long as it is grabbing things. Regarding recognition models, I guess most of you are aware of these two different ways of thinking about it, either induced fit or conformational selection. You start with a disordered protein and its partner. So if you go about with the induced fit mechanism, first you bind, and it is the interaction enthalpy that actually allows you to fold the partner. On the other hand, if you have the conformational selection model, first you order and then you bind. And it is this particularly ordered structure that we call residual structure in the ensemble. In the story I'm going to tell you about shortly, this is the model we adopt, and this is mainly by necessity because we have no idea who the partner is. Now, our favorite protein is called escargot, and we came across it because a colleague of mine, Dr. Veronica Narvaez at the Developmental Biology Lab, working at the same research center where I am, came across some mutants in this protein that impair the development of the larvae of the fruit fly. This is a normal fly, and you can tell that having low levels of this protein really make it look hideous. It is very important in all multicellular animals, which is basically animals. Otherwise, if they're unicellular, they're called protozoa, so it doesn't matter. Anyway, she was actually studying sensitivity to nicotine. And it turns out that depending on the levels of escargot that you can get the flies to be insensitive to nicotine. And it's important, it is a cute model to study nicotine addiction. In a more general context, it is important for the development of animals in general in order to get different layers of tissue. Oh, okay, fine. So escargot is a protein that belongs to the snail family. And like I said, it appears in multicellular animals. Escargot and all the members of the family are transcription factors. These are proteins that are in charge of telling the transcription machinery of the cells which are the genes that should be expressed at a particular time. And in order to do that, most of them have to bind to DNA. That happens through the C-terminal region of the protein. And most members of this family have zinc fingers. So this part is very well understood for this protein family. On the other hand, the N-terminal half is very poorly characterized, and this is mainly because it is disordered. So there is no way to know what it looks like. We looked at for sequences that look like escargot in NCBI, and it turns out that escargot exists in multicellular animals. all the way from sponges to vertebrates, which are all the way down here. So we got sequences of escargot proteins from all these different animals and compared them. Turns out that their C-terminal domain, the part with the zinc fingers, is similar between all of them, and that's what defines them as members of the family. But if you try to align the N-terminal region, it's impossible. And that is a hallmark of intrinsically disordered regions. Sequence is very poorly conserved, but composition is not. Okay? Now, we calculated, and by the way, when I say we, I mean Teresa Hernandez, and she's, what I'm telling you about is her PhD thesis. How disorder prone is the whole escargot protein for Drosophila and for all these animals that I told you about. So this is a consensus disorder prediction. We tried the 4 best predictors in the market as of 2020. And it turns out that if you go from the N-terminus to the C-terminus of the protein, this is the region of the zinc fingers. Anything below this dotted line tends to be ordered, and anything above it is disordered. So even away from the C-terminal zinc finger region, there is this small region, which we call S2, that is ordered prone. And for most of the animals that we tested, this pattern happens for many of them. So again, there is very poor conservation in sequence, but there is a conservation in the existence of an ordered prone region in the middle of a sea of disorder in the protein. Now, according to Sider, which is a predictor from, I missed his name. Never mind, it will come back from me. The idea from Sider is that it calculates the number of charges, of negative and positive charges in a particular region of a protein. You can classify them. In this lower corner, you see very few charges. This is an increase in negative charges and increase in positive charges. So most charged balanced proteins go along this diagonal. Here you have polyanions, here you have polycations. And each of these regions have different conformations that are preferred by them. So most of the S2 regions of the animals that we tested, which are listed here, tend to lie in this very compact prone region. And that is because this region is rich in aromatic residues, hydrophobic residues, and PST residues, that is proline, serine, and threonine, and these are disorder-prone residues. Also, this is a signature for phosphorylatable regions, and that is important because phosphorylation will include charges, and that will change the physicochemical behavior of this protein. The only way in which you get a reasonable sequence alignment of the S2 region, which is what is boxed here in red, is if you circumscribe yourself to flies. If you include also mosquitoes, then the alignment goes haywire. So it appears that throughout evolution, what mattered about this region is that it would be prone to being compact. And that's about it. Now, going back to Drosophila, because this is the actual experimental model that we can work with, what Tere did was to take this particular order-prone region, the S2 region, and to give it to these 5 structure predictors. As you might imagine, there are no known structural proteins with any similar sequence to this region, so the predictions are really poor. I would not use them for anything serious except as seeds for molecular dynamic simulations. So what I want you to look at, this is colored from the M terminus to the C terminus, is that there is a tendency, first, for helical regions, and especially near the C terminus, there almost always tends to be an alpha helix. So this looks like an alpha morph. Okay. So what we would like to know is whether this is foldable in a stable fashion, or is it starts as an alpha helix and then all hell breaks loose and we lose it. How stable is this helix? If so, what stabilizes it? Could it be an alpha morph, and can we measure this experimentally? Our tool were MD simulations with NAMD, CHARM36, and the GBSA implicit model solvent implemented in NAMD. This is NAMD 12 at the time we started this. To validate our runs, following the suggestion of Michael Feig, we carried out simulations of the aqua peptide. We did 2 simulations of 16 microseconds each. I want to show you that, okay, this is time and this is the fraction of helix that we find calculated every 10 nanoseconds. My apologies for the slides in Spanish, didn't have time to translate them. So what you see is that we started from a perfect helix. Okay, so the helix breaks and then forms and breaks and forms and breaks and forms. But the 2 simulations have very different behavior. So one moral of this story is that we first carried out simulation 2, and we were ready to call it quits because the amount of helix in these simulations is way over 50%, and we know experimentally that it should not be over 20 or way over 20. But then we carried out this other simulation, and this guy could definitely break helix and make it again, but then there were some really long simulation times with no helix at all. So run many copies. That is the basic idea. And this is just to show you that we can get spaghettis and we can also get very nice alpha helices. And this is the percentage of time that you find each of the residues as a helix, and as you would expect, it frays from the edges and tends to be more stable in the center. Now that we believe that our description of helices is going to be okay with the force field that we're using, we ran each of the 27 models that Tere generated during 2 microseconds. 2 microseconds is not heroic, but 2 microseconds times 27 gives you 54 microseconds of sampling, which is not terribly bad. So what we did is to compare the structures sampled by each of the runs, everybody against everybody. Okay, so what I'm showing you is the comparison of pairwise root mean square deviations of structures sampled from a simulation called SP8, which starts like this, and SP9, which starts like this. Okay, so here is time for SP8, and this is time for SP9. Each of the diagonals corresponds to the comparison of the structures sampled within one of the simulations. So I believe this is SP8 and this is SP9. And what is interesting is what happens off the diagonals. So these 2 structures can be really different from each other. The highest RMSD that they had was almost 15 angstrom. But also they can converge to the point of being basically indistinguishable from each other. An RMSD of 2.55 that's the same structure. And this was not just for this pair of residues, it happened for a bunch of other too. So even though it is not really long simulations, it works. So what do we see? So again, this is percent of helicity, and this is the number of residues. Our initial ensemble had a lot of helix at the C terminus and a little bit at the N terminus. And after we run for 54 microseconds, this helix melts, but this guy remains. Okay, and that caught our attention. This is a contact map. If you see anything that is different from white, then it means that that contact between a particular pair of residues happened along the simulations. And these are hydrogen bonds. Of course, not everybody can make hydrogen bonds with their side chains, and that's why you see a bunch of white things over here. What we see boxed in both these plots is this particular alpha helix, and it is stabilized by a very nice ionic bridge between this arginine and this glutamate, and that's what's marked here. So, what do you do when you find the structure? You break it. So, we chose 4 different starting models with very different structures, and we attempted all of these mutations, starting in these 4 different structures, each of them simulated during 6 microseconds each of the models, so 24 microseconds sampling for each of them. And what I'm showing you here is, again, residue number against helicity. What is in black is the wild type sequence, and what is in red is the effect of the mutation. And here, where it says promedio, that should be average. That is the average helicity for the wild type and the mutant. And what I want you to see is that we didn't break the helices. Full stop. I mean, between 29 and 30, it's basically the same thing. The most interesting one is this guy. Valines are supposed to be helix breakers because this is a beta branch residue and it is entropically disfavorable for it to be in an alpha helix. But it's actually stabilized. So we thought, ew, this is again a problem of GPSA. We are unbalancing the description of hydrophobics versus hydrophilics. And no, because when we look for contacts in aqua, we see no increased long-distance contacts between hydrophobic residues. And aqua, despite having glutamines, is basically 80% carbons. So it's not a problem of the force field. What happens is that we have long-range interactions that stabilize the borders of the helices, and this happens through the interactions of aromatics with the hydrophobics that we included. So, what do you do after that? We try to compare 2 experiments. There are no experiments for this part of the protein reported anywhere. So, we convinced our collaborators to synthesize a smaller region of this 45 amino acid S2 region. So they made a 33 amino acid peptide, and we have the wild type version, a mutation to leucine, which is supposed to break the salt bridge, and a mutation to proline. Proline is a helix breaker, last time we checked in all biochemistry textbooks. So now we're using CHARM36M and TIP3P, that is the special TIP3 water model that comes with CHARM36M. And this M is supposed to mean that it was made especially for the simulation of IDPs. And these are all atom simulations done with Chromax and with mass repartición de masas. Anyway, something that allows us to use a 4 femtosecond time step instead of a 2 femtosecond time step. We started from a perfect helix and allowed the simulations to run until they forgot that they had started from a helix for at least 500 nanoseconds. And this is the fraction of alpha helix as a function of time for wild type, leucine, and proline. The simulations have not reached the same level of maturity, but at least now they're doing what they should be doing. The wild type has a little bit more helix than the leucine and more helix than the proline. So there we're doing good. But when we look at the amount of helicity as a function of sequence, I want you to notice, please, that we have, for example, for the wild type peptide, we have a lot of helix in a region that is plagued with prolines. Prolines are supposed to break helices. So if anybody can explain to me what is going on here, I would be really, really happy. And also for the leucine mutant, that one really read the textbook and understands that where there are prolines, there should not be helices, but it refuses to break this helix despite having lost the possibility to hydrogen bond to this arginine. So what we think is going on is that, again, we have long-range contacts that are stabilizing the borders of the helices, and that is what is preventing the helices from fraying. Okay? And if we try to make pictures of what is going on, then you can actually see the helices and you can see the long-range contacts, especially between hydrophobic residues and the aromatic residues. So these are happening both in the implicit solvent and the explicit solvent simulations. So in order to wrap this up, these are the questions that we wanted to answer, and this is what we can say so far. So is this region foldable in a stable fashion, or is it an IDR? From our contact maps, which were yellow all over, meaning all possible combinations of contacts could be seen, it looks like an IDR, but it also likes to be an alpha helix every once in a while. Is this C-terminal helix stable? Yes, and it can even be resistant to helix breaking mutations, and this is due to tertiary contacts. Could it be an alpha morph? Maybe it is even two alpha morphs, because we have a helix where legally it should be a helix because there are no prolines, and then even in a proline but aromatic rich region, we also see a very, very stable alpha helix. And can any of these be measured experimentally? Yes. Lina Rivillas, from a collaborator of mine, she's a very good chemist, and she already synthesized the peptides, and they measured with circular dichroism in a lot of trifluoroethanol the helical content of the wild type, the leucine, and the proline mutants. And we have a qualitative agreement with the amount of helix that is present. The issue here is that we should heed our own advice. I told you with Aqua that it was imperative to start from different structures. So now what we're doing is that we're starting new simulations of these same peptides, but from structures that we saw in the simulations with implicit solvent. And hopefully with this new ensemble, we will have better statistics. If you want to look at the gory details of this story, all that I told you about the implicit solvent simulations was published last July in ACS Omega. And finally, thank you for listening to me. I am part of a consortium called Laboratorio de Dinámica de Proteínas. Carlos Amero does NMR. Lina Rivillas is the chemist that does peptide synthesis, and she's an excellent spectroscopist. And Rodrigo Razo is our medicinal chemist. And this is a collaboration with Veronica Narvaez because she's the fly expert. And these are the supercomputing centers in Mexico that we pollute with our simulations. So I'm open to questions, should you have any.
Okay, so first, thank you very much, Nina, for this really nice talk.
Are you still there?
Yeah, I'm here. So we have a few questions from, actually first is a comment from Hugo from Porto Alegre here in Brazil. So he's saying that, I don't know if you can read in the screen or not, but he said that this is not necessarily a break, but he understands that Pro may, in some cases, distort the alpha helix, and he suggests to use a tool to evaluate the geometry of the helix. So
yeah, actually, we are using Stride in order to measure alpha helix content because that tends to be the standard in the field. I will look at the tools in Chromax to see if we can measure it in other ways. Tere tried to measure not only helix breaking, but also bending, because we know that prolines can make helices. They are very frequent in hydrophobic environments like membrane, where there is no competition with water. It's not a problem of geometry. It's a problem of missing one hydrogen bond to stabilize the helix. So if there is no competition with water, then everything is okay, right? But the issue is that they should break because we're doing water. But I will look at ways to see what happens to the helices. Thank you.
okay, great. So now a question from Hugo. So he asked if instead of looking for the complete ensemble, have you tried to look for group of conformations? For example, identify grouped using PCA.
We have not tried PCA because this is a very diffusive ensemble. So from what I understand, PCA works when you are in a basin. So you're not moving very far from where you started. That may be a wrong preconception I have. So the problem is that this moves around a lot. If I try to get an average structure, I get something very strange. So, but I don't know. I've never tried PCA.
Also, we have a question from Pilar Cosio. Have you tried using enhanced sampling to calculate free energy profiles over a CV that measures helical content?
Our poor man's version of enhanced sampling is starting from from a bunch of structures. Many for us meant 27. Now it may mean 4 or 5. A collective variable, that would be nice, like to do metadynamics, I guess. The thing is that we do not want to force it into a particular structure. We want to know what it would like to do if left to its own devices. So I didn't show you, but we know that it likes to be an alpha helix, but you can also get it to be a beta sheet every once in a blue moon. So it's just that is so much less frequent than an alpha helix that it is very hard to measure. So if I use a collective variable that pushes it into an alpha helix, then maybe I'm biasing it instead of seeing what it can do on its own. I don't know. But there was, previous to that, there was something else that Hugo was saying that I missed. It's good to see you, Pilar.
Great. So we have another question now from Leandro Martinez. It is interesting in your case to study the stability of the conformations in different solvent mixtures, urea, for example. Can that be done experimentally?
Yes, it can. The nasty thing about these peptides is that they're a bit hydrophobic, so they tend to aggregate. Now, urea would promote denaturation. So what I would really like is to be able to do the simulations with trifluoroethanol, so that would bring it closer to the setting in which the percent of helix was being made, but I'm not aware of any field with decent parameters with trifluoroethanol. Also, my experience with urea, we tried to unfold for another project a small immunoglobulin domain with urea, and we blasted it with 10 molar urea and the thing would not budge. We had to increase the temperature, so then we had 2 perturbants, which was 8 molar or 10 molar urea, something that was already outside the normal solubility limit of urea. And maybe the problem is charm. I don't know. It might be an issue with the force field. But it would be beautiful. Now, again, if they measure the amount of helix without TFE, they don't get a signature in circular dichroism. So actually what they had to do is to take the lyophilized peptides, dissolve them in TFE, so 100% TFE, and then titulate, well, actually go adding a little bit of water at a time and seeing what happened with the spectrum. So these are really unstable helices, and that is what you expect from an IDR, by the way. I don't think that that is a problem, that it's behaving as it should.
Okay, so thank you very much, Nina, for the answer. So also Leandro just make a comment about the PCA, that they have some cluster strategy can work and help.
We tried. The problem is, which cutoff do you use for clustering? So from the 2D RMSD picture that I showed you, pairs of structures coming within 3 angstroms of each other are far and in between. They don't happen too often. And once you go beyond a cutoff of, say, 4 or 5 angstrom, it's very hard to say that they are the same thing. Very, very hard. So again, that's the problem. These are disordered proteins. They don't stay put. So most of the strategies that one could invoke for working with nice folded structures are not applicable here. But thank you.
Great. So, and then Leandro said that we'll send you an email, so you can talk about this a little bit. Pilar says it's nice to see you too.
Cool.
Yeah, and Hugo just want to provoke a little bit and say that he has better experience for the peptide with Gromos than with Char. So that.
Okay, which version of Chromos? Which force field?
Yeah, so.
Yeah, I'm willing to try. I mean, at this point, I'm using Charm 36M because it's supposed to be around the best thing next to freshly baked bread. But which version of Chromos?
Yeah, I wait Hugo answer, or I believe that.
Or if not, by email. We can talk to each other later.
Yeah. No, he just said that both beta sheets and helix even start from the broncho formations up to around 30 angstroms. And also a final question, so we can move on.
Sure.
Sorry guys, but there's a lot of questions. It's a really nice talk. So Matias Machado asked, have you tried to stabilize the beta sheet conformation instead of breaking the of a helix?
That would be nice, though I have no really good idea of how to do that. Maybe I could try to engineer a disulfide bond. But again, in the physiological context of this thing, this is an intracellular protein in a reducing environment, so disulfide bridges wouldn't work. I have to look at SP6, which is the beta-rich structure, and see which interactions are happening there. Curiously enough, those sheets melt. Okay, now you lost me.
Yeah, it's nice when it happens. This happens. At least I like, so you have to look for this. Also, I'll not make any questions. I will look your paper and send you an email because I'm really interested in how water behaves near distinct sites. And actually, we have now you discuss about the difference between using explicit or implicit water. And now I'm finishing a work with a guy from Mexico, really, Raul Fuentes. He is now in the University of the Universidad Autónoma de México, the Ciudad de México.
Mm-hmm.
And we are studying how actually it's all atomic water, but we are including or excluding how the flexibility of the molecule change the behavior near neutral or charged surface. So we can talk a little bit and perhaps do something together. So
I'd love to.
Yeah, I'll send you an email and also include Raul in the discussion so we can make this Brazil and Mexico work.
I like that.
So yeah, so thank you very much, Nina, for your really, really nice talk. So now
I'll stop.
we'll go.
Yeah.
Yeah, I already stopped it. So if you want to say one last goodbye.
Okay, now ready to listen to the next one.
Okay, so thank you, Nina. Now I will you have Denys back. So you are muted, Denys. Okay.
Yeah, now I'm back. Thank you very much.
Yeah.
Sorry for the internet problem.
Yeah, it's everyone's calling out the new normal, right? So I will put your stream so you can share. You can just include your.
Okay, I'm going to continue from. I think I have stopped. So I think is in this point here. So
yeah, well, something like this.
Yeah, exactly. So I have shown that Suave is able to calculate to build a surface, a fitting surface for compact and for open surfaces where independently of the shape of these surfaces. So once you have this type of function, and with this is strongly tested because we have tested with different functions in different surfaces, we can apply for different systems to see how this new analysis can show us differently the response of the system in relation to something that is not being calculated from analysis that are not taking into account the morphology. So Suave is able to calculate some of these, all of these systems that I'm showing here, but actually much more than this. So I'm just showing you applications for flat membranes, curved membranes, and for compact systems like the compact interfaces, like vesicles or micelles. But Suave is even able to calculate liquid-liquid interfaces properties, like, for example, the area of this liquid-liquid interface, the geometry of this interface, and so on. So I'm just showing some of the applications Suave is able to do with these interfaces, but Suave is able to calculate much more than this. So I'm going to start with the density profile calculation. That is one of the most common evaluations we do for molecular dynamic simulations for bilayers. And with this, I'm comparing the usual slicing method and the morphology-dependent slicing method that are implemented on usual tools and Suave. So when we are applying the usual slicing method, we are just making flat slices on the systems independent from the type of the surface. If the surface is curved or not, independently of this, we are making just flat slices along the z-axis of the system. That is normally the normal axis of the membrane. But when we are introducing a morphology-dependent slicing method, the slice have the same shape as the interface. So for flat membranes, this is not a problem, as you can see here, because the comparison between the two profiles, the density profile from usual slicing method and the morphology-dependent slicing method are very similar. And this is exactly because in this moment, when we have a flat membrane, the slices that are dependent, the morphology-dependent slicing method will create flat slices because the system is flat. But when we go to a curved membrane, this is not exactly what is happening. when we try to analyze the density profile for curved membranes using the usual slicing method, you'll see that probably we fail, because we are now trying to calculate something that is curved but making flat slices. And as you can see here, the conclusion from the first figure, that is the usual slicing method picture, you can see that that is not exactly well structured by layer, and that is water inside this, and you cannot see a very well localized, good distribution of the calcium ions and the phosphates for these systems. But this is not exactly what is happening in this system. When we just calculate exactly the same analysis, the density profile, but taking into account the morphology, using the morphology-dependent slicing method, you'll see that the membrane is well structured. And as you can see, there is no water inside this membrane, and you can see exactly the places where the calcium and the phosphates are. So the conclusions from these two analyses, just taking these two different perspectives, are very different. So once we are using these two different methods with a flat membrane, it's perfect, the results are similar. But when we are trying to analyze curved membranes, or even with a small curved membranes, or curved interface, chemical interface, you will see that the conclusions are completely different. And that's a problem if you are using this type of conclusions in your paper, or to analyze your system to see what is happening to your system during a simulation. So this is one of the applications we can see how the morphology-dependent slicing method, or how taking into account the curvature of the system and the morphology of the systems, can change the conclusions we are making from our system. But now I'm going to show you some of the other applications SWAB is able to do. So here I'm just showing you a bidimensional map, that is coloring map, that is showing us the curvature and how the curvature changes along the dimension of the bilayer. So as you can see in the picture C, for a flat bilayer, the distribution is like an homogeneous color. So this is expected because there is not curvature in this system. But when we go to a system like D, when we have a small curvature in the center of the membrane, we can see a variation, a gradient in the coloring of red. And this is exactly where we can see the variation in the structure we are seeing at right. And with this, we can see exactly where the curvature is being formed during our simulation, and exactly the intensity of this curvature. As you can see, for example, in the picture B, we can see exactly the quantity of curvature is being formed during the simulation for the systems, and exactly where this curvature is being formed. For the system I have shown in the second slide, I think, that is the interaction between microbial peptides and the gram-negative outer membranes of Pseudomonas aeruginosa. It was possible in that study to see exactly the place where the antimicrobial peptides were acting on the membrane because of this coloring map, because we could see exactly where the curvature was changing. And this was very nice because we could see exactly what was happening to the systems along the simulation and how much curvature was being formed during the simulation. So that's nice. But in this picture, we can see just a coloring map. We can use this type of analysis to calculate numbers. So here is one expression of these numbers that can show us and how the membrane is curved. This picture is the graphic for the distribution of deflection angle between the system normal vector, that is normally the z-axis of the system, and the surface normal vector. And with this, when you have a flat membrane, you see that the deflection angle between the surface normal vector and the system normal vector is approaching 0 degrees. But when we are starting to curve our membrane and start to have the curvature formation, you'll see a distribution in the deflection. And as you can see here, for example, in the picture in red, you can see that we can come from 0 degrees up to 60 degrees in curvature, in deflection angle between the system normal vector and the surface normal vector. And this characterizes a curved membrane. But when we see the same representation for the black, blue, and green, we can see this data for slightly curved membranes. And as you can see in the green system, there is a profile with the average angle in the deflection angle close to 10 degrees. This is just because of the normal fluctuation of the membrane along the simulation time. So this represents and this characterizes a very flat system just fluctuating a long time. So these are just one of the applications we can make for flat systems. We can make much more than this, but I'm just going to show this for the open ones, and I'm going to show you some of the applications for the closed surfaces. So this is a micelle, a DTATF micelle. We have a simulation, a very short simulation for this. And in these simulations, we are able to calculate the fitting surface, as you can see here in green. We are trying to fit the red points, and with this curved surface, we can calculate the area of this micelle, the total volume of this micelle, the average radius, and we can even calculate the moment of inertia of this micelle, the principal axis of inertia. And this is quite nice because we can actually see exactly the shape of this micelle according to the moment of inertia, the principal axis of inertia. So that's one of the applications that are nice to compare, because you can compare exactly the result from the simulations to what you see in experiments. You can see the shape of the micelle in simulation and the shape in the experiments, and you can compare them directly just because we have used the shape of the surface fitted by SUAVE. And we can go to a more complex systems like a lipid A vesicle. So in this vesicle, we can make two surfaces, one external and one internal surface. With the external surface, we are using the phosphate groups from the outer leaflet of this vesicle, and the internal surface, we are trying to fit the phosphate groups from the inner leaflet of this lipid vesicle. And with this, it's very interesting to see, for example, some of the properties are calculated for 100 milliseconds for these systems. As you can see, for this system, the external surface is increasing along the simulation. And with this, the volume of the total spherical shell volume, that is the volume comprised by the lipid region, is also increasing. And this is very interesting because when we see the average radius for this external and the internal surfaces we are fitting with SUAVE, you can see that the external surface is increasing. As you can see, the volume is increasing, so the external surface is increasing, but the internal one is decreasing. So that's very interesting to see that one of the interfaces is not increasing, it's just decreasing. And this implies that the thickness between these two surfaces is also increasing, as you can see in this picture here. So we can see exactly what is going on with this vesicle a long time, just because we are using the interface built fitted by SUAVE. And you can actually do something that is not so common for most of the programs that we can see nowadays. That is the density profile for a closed surface like this. So we can see here a region where you have the lipids for this spherical shell, and then you can see water in the inner region of the spherical shell and water outside, but there's no water, reasonable amount of water inside the lipid, so the lipid region. So you can see that this vesicle is well structured, and you can see that we can actually tell exactly what is going on with the systems, just because we are now able to calculate what is going on with the surfaces, because we have used the fitted surface to do so. So these are just some of the applications SUAVE is able to use to extract information from the simulations, but we can actually do much more than this. SUAVE is, in few words, a program that calculates geometrical and physical properties of any type of chemical interface, taking into account its morphology. It's free for download, so if you want, just go to biomathsite.net/SUAVE software, and you can actually email me, because I will be there to answer any question. And just one of the applications you can do with SUAVE, you can calculate the area and the volume per molecule, the membrane or for a vesicle thickness, the density profile, curvature of the parameters, Gaussian and mean curvature for the systems, level surfaces, and angular deflection distribution. We can even calculate the moment of inertia and other applications that I'm not listing here, but you can see in the website, SUAVE website. And to finish, I'd like to thank all the funding agencies that have made this dream come true. And then I'd like to thank my co-supervisor, Kaline Coutinho, and my supervisor, Professor Teresa Soares. And that's all. I think that now I have, thank to God, finished the presentation. Sorry for the internet connection.
Great. So thank you very much, Denys, for your really nice talk. And now, yeah, we have some problems, but this happens in this time. So just see if you have any questions from the audience. Right now, I have one question, but or a suggestion of work. I don't know. Have you tried to see what happens with this vesicle when you compress it, or just or you just look for it in the bulk and in equilibrium?
Yeah, in this simulation, this very new simulation we are doing, we didn't try to compress the vesicle. We just put in a bulk solution to see what happens because we are equilibrating this one. So as you could see, the area is increasing too much in a very short time. So this is exactly because this is the equilibration step. So during these equilibrations, it's nice to see what is happening to the vesicle. So this is why I have shown these figures. But it's a nice idea to put compression to see if the area per lipid is compressing and is decreasing. So it's one of the ideas. Once I have the tools to do such an analysis is possible to do anything. So once you do something to your simulation, it's possible to see exactly what is going on with the area or the thickness, for example. It's a nice idea to make it.
I have because it's not only compression, but we are studying when you put a bubble-like cell, it's not like exactly a cell, but it's more like a bubble, and you try to move it to a polymeric network. So you can have some attractive sites, this kind of stuff, and I believe that you can start with this just a vesicle to see how it gets stretched by the polymer or this kind of stuff. But we can talk later
That's a nice idea.
about
That's a
this.
nice idea.
Yeah, because I'm really interested in this, and your software will probably help me in the future. So now I have a question from Sergio from Uruguay. So he's a great talk,
Okay.
and
Hi, Pedro.
and he has how flexible is it with input formats, and can you adapt Suave on coarse grain trajectories?
Yeah, actually, we can do that. For example, we have chosen the PDB format not because it's just common in some programs, but because it's a well-established pattern. For example, if you are a user for GROMACS, normally you'll have a TRR file for the trajectory. So, but not all the people use GROMACS. So some people use different codes and different trajectory files. So we have used the PDB format because it's more general. So once you have a PDB format, whenever the type of simulation, if you are using the coarse grain, if you are using all atom, if you are not using atom, like if you are using really huge beads in a coarse grain model, coarse grain model, you see that you can just insert the position of the atoms or the beads for the coarse grain model. Even I have not presented here, but I have this type of analysis for vesicles made in coarse grain. So once I have the coarse grain models, if I have the position in space of the atoms or beads, I can calculate anything you want. So I can make exactly what I have shown here for atoms. I can calculate the fitting surface for coarse grain beads as well. So this is one of the applications. So the input format should just must be in PDB format. So we just have to make a conversion from, I don't know, different types of TRR trajectory files to PDB format. And once you have this, you can calculate whatever you want. And just one clue, I think, is when we are calculating, for example, a system and you want to make a trajectory in PDB file, normally this PDB file is heavier in the system. So you can use it, for example, in the systems that I have shown here, I have done the PDB trajectories just with the phosphate groups, because I was going to calculate just with the phosphate group. So I didn't need the all atoms if I just need the phosphate groups to calculate the interface. So once you make the trajectory just with the atoms that are important for you to make the analysis, you can do all of the analysis Suave is able to do.
Okay, so thank you. Now another question from Hugo. So how would Suave behave in protein surfaces as a faster way to detect pockets for virtual screening?
Yeah, that's a nice question. Actually, I think I haven't tried to proteins this type of, for example, the closed Suave. The closed is the spherical one, the spherical implementation of Suave. But it's possible once you have the atoms you want to use for as indexes for the fitting procedure, you can have the mapping of the surface. The problem is just having these points where you can tell exactly where the surface has to pass. So once you can exactly mark the points in the surface, points in the protein you want to use for Suave to make the fitting process, you can calculate whenever you want. The problem is just to mark this, to select these atoms. This is just one of the problems, but we have an application made in Suave that is the S-index, and this S-index program will tell you exactly the points you probably will need to calculate some properties, and it will make the task of making these index files easier than if you are trying just by hand. So you can do that by Suave, making the index files and try to make it easy for a protein. I've also done these for example, MOFs and for vesicles and micelles. So closed surfaces are perfect to calculate with Suave. The problem is just when you have to select the atoms you want the surface to take into account when making the process of fitting. Just this.
Thank you. So also, here's the answer from Hugo.
Yeah. Actually, we have not published implementation for Suave that is exactly made for this, to calculate the cavities into some structures. We have done this for MOFs, for example, but we can do this for proteins. If you use the whole number of atoms, the whole particles of a protein, for example, you can see exactly the spaces and cavities into this protein. So it's not published yet, but we are now writing the paper to publish this version of Suave, but it's possible to do that. Once you have all atoms of the proteins and you have the van der Waals radii for these atoms, you can calculate the cavities from this inside this protein or inside any structure. We have done this for MOFs, and it's quite nice to see the results, because you can see exactly the space into the MOFs that you can use to calculate, to insert some chemicals, or to understand the transport of mass by the MOFs according to the space, the free space inside the MOFs. So the proteins can do exactly the same.
Okay, great. And Leandro has a suggestion. The package name chem files which is compatible with many types of trajectories with which you can use to avoid the user having to do conversions.
Oh, so that's nice. I didn't hear about this, so thank you very much for this suggestion. I'll take a look on this to see if we can implement on Suave how to ease this task of transferring one type of trajectory to another type of trajectory. So thank you very much, Leandro. I'll take a look on this.
Okay, guys. So Denys, I want to thank you very much for your talk. I also have another comment saying that it was a great presentation.
Thank you very much.
Thank you very much. Thank you, Nina. Thank to everyone that watched this live webinar. And so see you guys in 3 or 4 weeks in our last seminar of this year. So thank you, Denys.
Thank you. Bye-bye.
Bye, guys.